and Garba.ai, which is in the space of healthcare. And I'm also chairperson for AI framework implementation with ITS India Forum. So I closely work with Indian government. And like ma'am has said, I'm a visiting faculty at ISB. I hold a double master's degree from IIT and ISB. I work with HSBC, ITC, Infotech, Infosys, and Deloitte. And today, as we speak, for the last 7 years, I've been delivering trainings in the space of AI and Gen AI and Agentic AI of late. All right, so let's get started. I would like to first talk about a few of the interesting projects, just to give you all a gist, right, on what kind of work is happening in the space of life sciences healthcare. Then I'll switch to our subject, okay, and then talk more about that. And I'm hoping that there'll be some value add throughout. Let's see. So yeah, we have 100 plus people working across. There are a lot of projects which got published on international journals, including London Business School Journal. There are 250 plus journal publications that we have. And there are 10 plus governments for whom we do projects. All that is marketing, isn't that right? So we do projects for different customers, including Biologically Limited, Dr. Reddy's, and we are working with another company called as Komen, which is into healthcare device manufacturing. I'll get to that after I talk about this. So this is one of the solutions in the space of AI where people who don't have kids, naturally, they go for treatment called as in vitro fertilization. Then you take sperm from male, oocyte from female, you fertilize outside to form embryo. This embryo you grow in the lab setting, and embryologist would observe this particular growth of embryo under the microscope. So device is coming into picture, microscopic device. And with a lot of subjective belief, embryologists will implant embryo into the woman's womb. And woman will then realize whether she is pregnant or not after 2 months. And the success rate is only mere 35% across the globe. So in most likely case, woman's womb is cleaned and cycle 2 repeats. So we have built a solution where the moment you upload embryo images, AI will actually give you a grading on whether embryo is of grade A or B or C, alongside building explainability. Okay, that's called as GradCam based explainability, gradient class activation map. So that's one of the solutions. Then Gen AI solutions, right? So we have taken a medical large language model. Typically, whenever you want to build a large language model, whenever you want to build a Gen AI solution using large language model, and if you want to do that in the space of medical, you need to use medical large language model. You cannot use the typical large language models which people have. Okay. And to quickly give you a gist of this, I would like to take your attention to this presentation. Always, because everybody is talking about Gen AI nowadays, you need to first do research and understand what are the medical large language models which are trained on medical literature. Okay, you cannot take anything and everything because they are trained on internet data. The other models, such as GPT model from OpenAI, etc., they are trained on generic data, not deep medical literature. So you will have to do this research and then build a Gen AI solution on top of that, right? And specifically for radiology, specifically for a few scenarios, people have built such models. And this is going to be the future. Micro specialized language models, which will be solving very, very specific needs. Of course, generic is something which will attract the mass market, like ChatGPT, everybody must be using now. But domain specific models will cater to a small segment, but to a niche segment. And that's on rise now. Alongside this, another thing, another solution which is on rise is your edge AI. That is going to take us to the next discussion. In 2024, if I'm not mistaken, I visited China, right? Why did I do so? Because there's a company called as Comen, Comen, and you would see their devices in ICUs, okay, intensive care units, where you'll have a lot of these kind of devices which capture the vitals of a patient. And what typically happens is, if these vitals, which are being collected by the patients, if they fall outside the limits, outside the control limits or specification limits, it will give a beep. And the person who is in the ICU could be doctor or could be nursing administration staff. They'll go to that specific patient, they will see what's going wrong, and then they'll just let that buzzer go away. In most cases, or in some cases, they will do some intervention to revive the patient. However, in ICU, if a patient is in ICU, then that in itself means that patient is critical. On top of that, if you're just looking at some buzzer, if the values are outside the range, then if you go and try to, attend to the patient, would it be helpful? Not really, right? So we started collecting the data from Komen, the patient vital details. And we also captured the details on what happens to the patient. Did the patient experience sepsis attack? Did the patient experience heart attack? Did the patient experience brain stroke? What did the patient actually experience? This information we started collecting. But for how long should we wait? Should we wait for multiple patients to experience that sepsis attack or brain stroke, etc., and then start collecting the data and then build your model? Then we thought, no, this is not the ideal way out. So we built clinical practice guidelines. We have taken it from the medical, council, and then we looked at what are the different vitals, in which way vitals, if vitals behave in a specific manner, then what kind of outcome patient might experience. We looked at all of these things and we built a system. And we also ensured that only doctors who are qualified with a username and password can enter into that particular system and look at the predictions. Because not everybody should be given access to that. Only the doctors were given access. And there, based on the vitals, some indications were being given that patient might experience this problem, patient might experience that problem. But this is more of reactive, isn't it? Patient is experiencing some problem and you're saying because the heart rate is high, blood pressure is low, patient might get sepsis attack. That is more or less reactive. What is AI there? So we improvise a solution a bit, and this is what we built. And these are okay. We built a system where our solution will capture heart rate, respiratory rate, blood pressure, dialytic blood pressure, oxygen supply, temperature, all these parameters. And it will also do something called as forecasting. That means, given all the vitals of a specific patient, of course, it has to, uh, you know, continue for a few specific seconds. Then you get that historical data. And based on that, you forecast. We forecast the heart rate, respiratory rate on all of these metrics. And after we forecast all these, then we predict. Then we predict whether someone will have some kind of a sepsis attack, or what will be the critical, clinical interventions which will be needed, so on and so forth, right? This is what we have built. Of course, I just took the video of the solution when it was in pilot phase long, long back. I'm not allowed to show you the actual minimum viable solution which the client is using today. All right. Now I'll walk you through the discussion in slightly more detail. But do we have any questions for me, friends, until now? Faculty members, doctors, do you have any questions for me? All good? Can I go ahead?
No such questions, sir. You
Yeah.
can.
Okay, fine. Biomedical signals, right? You have ECG, you have EEG, you have EMG, so on and so forth. Is anyone aware of which of these signals is used for what purposes? What are those? Can you help me understand? Yes, ECG is for heart, absolutely. Then EEG is for brain. Then EMG is for muscles. Wonderful. Yeah, good. Huh. Even before okay, fine. Just remind me if I forget. I would like to talk about agentic AI solution also, because one of the questions is which area inside the hospital AI is playing a vital role. And you have connected with the name called admin. So okay, let me show. Yeah, let me show quickly. Let me launch a specific application here. Yeah, wonderful. Agentic AI, a lot of people are talking about that, right? The way it works is, whenever a patient walks in, patient will fill the form, or the people at the billing counter will fill the form. And then you enter the details of patients into the database automatically. Whatever application form on mobile or on computer, when they fill it, the details are entered into the database. From there, you check whether the patient should go to the emergency route, or should the patient go to OPD. Right? This is one workflow. The moment you go to OPD, doctor has to be scheduled, doctor's appointment. And in this doctor details database, you'll have details of all the doctors. Then once you get the details of all those doctors, right, like this, you extract the doctor details. Then after your consultation is done, there'll be a prescription. That's called as prescription agent. Okay. What does the prescription agent do? Prescription agent will automatically take you to the lab test routine. If there are certain lab tests that have to be conducted, it will take you to another lab agent. If not, you'll go to the pharmacy agent. Then, if you go to pharmacy agent will look at what are the medicines and what are the prices of those medicines, right? These details will be retrieved. If you go to, say, emergency department, instead of OPD, doctor consultation, here emergency agent will look at who are the emergency doctors who are available. And then since it is emergency case, you might most probably go to ICU. So it might also become important to check whether you have insurance or not. So the insurance agent will kick in. Okay. And the lab agent will look at what are the lab prices, etc. Okay. And then create a bill for you. And ultimately, you'll have billing agent which will generate the cost of the treatment, cost of your lab prices, etc. And then it will also trigger an insurance agent, and this insurance agent will check whether you are eligible for insurance or not. Okay. And this is how you can use AI inside your hospital for multiple purposes, and you can automate the entire flow, right? All these reports will be generated like that. So for the question that our friend had, which area, across all the areas you can use AI, but this is one example. Directly, do we have any AI tool, Himanshu? I'm not aware of. Maybe there are, but unfortunately, I'm not aware of those. Okay, let me, yeah, let this be like that. And if you want to read more about our solution Garba, you go to aispry.ai. In products, you'll see Garba. And there's also a detailed explanation on what are the different elements that we are addressing, from sperm quality assessment to oocyte quality to ERA to PGT and embryo grading that I've shown you. Everything is available here, right? You can go through that whenever you find time. Or you can go to projects, and under projects, you'll see huge number of projects, and you'll have to pick up what are the interesting areas for you, and then you can explore further. Yeah. Back to our discussion. So we have ECG, EEG, EMG, right? Why biomedical signal processing? Why do we need it now? So the key insight is every second, our body generates 11 million bits of sensory information. Yet, our conscious mind processes only 50 bits. So, biomedical signal processing creates a translator between the body's electrical language and actionable clinical insights. The question to you all is, why did it take until the 21st century for AI to impact biomedical signal analysis? And do you know when ECG was invented, right? 1903. But why do you think AI was not efficiently used in this field until 21st century? What are your thoughts? Anyone? Feel free to unmute and answer as well. Nitya, good. Selva, absolutely. Computational power. Where was it? There was no computational power, right? Second thing is digitization. For your signals to move from analog to digital, it took a lot of time. Then data availability also was an issue. And the real breakthrough came in when deep learning algorithms came in. Instead of manually, looking at all the, features, variables whose data was getting generated, and then manually thinking about which variable should I combine with another variable to come up with a new feature. That's manual feature construction. Because you'll have huge amount of data coming your way, understanding and establishing the correlations was taking immense amount of time. So deep learning has come into picture and it has eliminated the manual feature engineering process and it automated the feature engineering process. Of course, it is black box, but results are good. All right, that's right thing. And second thing, if biomedical signals contain so much diagnostic information, why can't AI replace clinical judgment entirely, friends? Why? Why can't I have AI take over completely? Because biomedical signals are giving a lot of diagnostic information on what is happening to the patient. AI can read all these patterns. And then can it not completely replace the clinical judgment? If no, why? AI is very. What if we build a model with 99.9% accuracy, Nitya? Because you have humongous amount of data. You're right, right? AI excels at pattern recognition within defined parameters. Whatever heart rate or your blood pressure, oxygen level, all these, if they are in the defined parameters within a specific range, then AI does very good job. But for medicine, we need the other context also, such as was the patient having some anxiety? What are the cultural factors? Is a patient using some medication? All of these things are extremely important. For example, AI might detect AFib with 99% accuracy. Even in Apple Watches, you have that. But deciding on anti um, coagulation, it requires understanding fall risk and quality of life goals. So a lot of things have to be understood. All right. So here we have ECG, EEG, EMG, three languages of the body. Right? And biomedical signals are not just data. It's an attempt of your body trying to communicate with the different other parts, right? Um, so the signal is a symptom, right? Not a disease. Now, ECG, EEG, and EMG all measure electrical activity. Why can't we have a universal algorithm? One algorithm for all the purposes. Why do we need different algorithms, machine learning algorithms, etc.? Because each of these signals, now you should not say that I'll combine all of these and build one model. It is not going to give you beneficial results. It's like mixing apples with oranges. And we know that AI solutions, if they are built on homogeneous data, they do a good job. So they differ on amplitude, frequency, sources, and clinical meaning, so on and so forth. So you cannot combine them all together, right? Okay. If EEG signals come from billions of neurons, How can we extract meaningful information from chaos? How would you be able to extract some meaningful information? You can extract using a phenomenon called as neural synchronization. Individual neurons, they keep firing chaotically. Okay. And what EEG does is it will apply a technique called as neural synchronization. Deep sleep means millions fire together. There is some specific wave pattern, right, which goes on. If you're deeply concentrating on something, desynchronization happens. Beta waves are captured, right? Ultimately, EEG captures collective behavior. All right, collective. So that's how the analogy is. You hear the sound of the entire stadium if you watch a cricket match, not every individual's voice, right? So that's an example. Okay, next. Let's look at ECG, right? The heart's electrical signature. The simple ECG waveform is actually millions of cardiac cells depolarizing in coordinated waves. A 1 millisecond delay can mean life or death. Just 1 millisecond delay. I'm a heart patient. Yeah, I understand these things. So AI measures with microsecond precision and never gets tired. So AI will continuously keep analyzing this, right? So there is something called as the QRS complex, which lasts only 80 to 120 milliseconds. Why is such a brief event critical, clinically critical? Because we are talking about analyzing all these data points, data getting generated from each of these. Okay. So if QRS value, if it is greater than 120 milliseconds, if it's slightly prolonged, it's called as conduction delay. If it's greater than 150, in heart failures, it is called as dysynchronous heart, where ventricles are not contracting together. Efficiency drops. You have something called as ejection uh, fraction, right? EF value. Um, so all these things I also learned, in 2022. Yeah, in 2022 Feb, wow, it's almost 4 years now. I experienced a major heart stroke. Rushed to hospital. Then they said that there is 100% block, and they put a stent, one stent, by the way. But all these things are very critical, right? Until unless one experiences, you might not be aware of how this data can be so critical. It's a difference between life and death, and so on and so forth. All right. Then this single measurement will determine eligibility for CRT, which of course you all know that it will be saving your life, right? Why use 12 leads when 3 points can technically triangulate the heart's electrical activity? So typically, okay, without getting into the finer detail here, I would like to tell you that whenever you want to build any AI solution on the data, whether it is coming from ECG or EEG or EMG, etc., you need to understand the domain aspects. Without having domain knowledge, building an AI solution is next to impossible. Either you need a domain expert or you need to learn on your own and gain that expertise before you build the model. Okay. EEG pertaining to brain, right? Brain operates on multiple frequencies. We know that. You have the right side on, you have prefrontal, so on and so forth. Okay. Now, there is something called as alpha waves, right? Typically, when you're idle, you say you're relaxed. And why do they disappear when we open our eyes or think hard? So alpha blocking reveals something profound, right? Alpha means idle neurons, standby. Okay. And then if the alpha value is reduced, it will increase your activity. And if the alpha value is enhanced, then it alerts your relaxation. So in this way, there are quite a few things even here. You have some gamma value, PCA value, so on and so forth. Same is the case with EMG also. Okay. In EMG also, you have a lot of data available. You need to understand the finer details on what is the difference between surface EMG, needle EMG, and bunch of things. And then you get into further analytics. Once you have all those signal, you will, and all these are time series based. In the video also I've shown that it's all time series based. If you see, continuously the data is getting generated, right? And since it is time series based, you can extract a lot of time related features, which can be useful ultimately for your AI solution building. The quality of the data will determine the quality of your model output. And in life sciences healthcare, the first preference should be given not to your deep learning models, but preference should be given to non deep learning models, right? Shallow machine learning models, because they have a lot of explainability, like regression techniques, logistic regression or linear regression or decision tree. These kind of models are preferred because they build in explainability. They will tell you why the output is predicted in a specific manner. What are the reasons? You'll come to know. But if you use deep learning, the accuracy of your model will be very good, but you cannot really explain the output. Okay. So simply put, if you can sweat on data preparation, ensuring that the data is of good quality, and then if you do proper feature engineering, then you can use the data and apply your simpler models which have explainability built in, right? As opposed to taking, a data set which is not that well processed. And if you don't do proper feature engineering, and then if you apply to deep learning model, it would be meaningless. Of course, deep learning models give you more accuracy. Okay. So you remove the noise, right? Based on, again, to remove the noise also, you need domain knowledge. You have to ensure that there is as much of signal as possible than noise. Okay. Filtering, if you just filter out certain things, it will remove noise. But how do you ensure that clinical information is not removed? For example, ECG info less than this. Okay, that's what your domain knowledge might say. So you can safely remove 60 hertz noise. Then you have something called as adaptive filtering. Then you have something called as validation studies. Then you have something called as transparency. All these techniques will help you get some clean data, more of signal, less of noise. Right? And deep learning learns features from raw signals. Traditional feature extraction obsolete? No, you can do it. But the challenge is when you traditionally perform feature engineering, like okay, I will take ECG information which is less than 40 hertz, greater than that, I'll remove it. You are coming up with this manual process, which is good, but time consuming. And there might be a lot of such features which human humans have not even seen, probably. A lot of such cleaning might be needed. In such scenarios, you can use deep learning models, which will automatically identify very, very complex patterns which you cannot even imagine, right? And how do we know that? Because the accuracy of deep learning models is very high. However, interpretation doesn't exist. So, to get interpretation, you need to certainly look at handcrafted features, manual feature extraction or feature engineering should happen. And then you know what are the features that you're working with. And then you'll have the interpretation built. Okay. The next big thing is edge AI in healthcare. What do you mean by edge AI? Whether you like it or do not, AI is coming closer to you. In the sense, I can just open the phone, I can look into it, and using my image analytics, it got unlocked. Right? So, edge AI is coming very, very close to us without our knowledge. Now, while my phone captures my face, do you think it is sending the information to the server somewhere? And from there, do you think the recognition, facial recognition is happening? And then it's sending message back to my mobile. And then is my phone getting unlocked? Not really. Even if you don't have internet connection, even if you don't have signals, mobile connectivity, it will still unlock because AI, the AI solution which will recognize my face is inside this device. That's called as edge AI. And the example of Garba that I've given is pertaining to this edge AI, right? So if you look at IVF microscopes, this is how the microscopes will look like. One way is to capture the microscopic image, pass it to your computer. From computer, you send it to the application, which is garba.ai, and then it will process and give you the predictions. This is one way to look at it. Okay. Second way to look at it is what if I reduce all this headache, put this particular AI solution inside the microscope so that the moment um, embryo is placed under the microscope, immediately it should do the prediction. And the prediction result should immediately show up. So the latency, the time taken to process and show the results will be faster, right? And also data privacy. You're not storing data anywhere. No images stored anywhere. Directly processing is happening. So the data privacy headache is also taken care of. Okay. Sorry. Okay. So that is what is edge AI, right? It's not just speed, it is democratization, meaning you need not send the data to cloud, you don't need internet connection, don't need data centers, no subscription needed. All we need to do is sell it to microscope, IVF microscope manufacturing company, our solution. They'll put our solution inside and they keep selling it. No headache. And edge devices, they work in remotest of the areas also, right? Where you don't have internet connectivity, etc. Rural areas, disaster zones, war zones. you know, so on and so forth. And you're making healthcare accessible to common people also who cannot afford premium services. So edge devices have limited compute though, right? How can you run complex AI that typically needs powerful GPUs? Any thoughts? If you want to process images, etc., you need a powerful computer. But when you say edge device, it could be on the mobile, and mobiles are not as powerful as the servers, right? And then how do I put this edge device? And how do I ensure that complex AI models also run there? Edge means you put this model inside that, Selvakumar, inside the device. For example, let me see if I can show you something. Let me go here, for example, and help you understand a bit. Okay. So we are doing a project for Northwestern Railways. In Northwestern Railways, we are given a task to monitor the people inside the cabin. Okay. And here the ask was, if pilot and co-pilot are missing, both of them missing means what if there is an accident? Who is to be blamed, right? So both of them are missing. And we wanted to, or we wanted to continuously monitor using video analytics solution. So we determine whether someone is present or not, someone is sleepy or not. Are they micro sleeping, meaning just for a brief period they'll jolt like that? That's called as micro sleeping. Are they micro sleeping or not? All these things typically, right? Now the question is, if the train is on the move, you know about the regions, right? In majority of the places, you won't get signal. So the question is, we cannot share or send this data to the server, process and send the message back. It will be too much of a time. So we use edge devices such as NVIDIA Jetson Nano Pro, Nano Orion, I think. Let's see. Yeah, NVIDIA Jetson Orion Nano. So these are like small computers. I'll put a camera to it of right configuration. I'll put my model in a in a micro SD card storage device, and I'll put it there. It's like a small computer. Okay, it's like a small computer. That's it. And then the processing will happen real time. That's the meaning of edge in this scenario. It's not that again you're sending data somewhere. Model will be inside the machines in ICU, etc. It will be inside. Okay, that's the point. Okay, hope I'm making sense, Silakumar. Okay, these are all the IoT devices inside which you put the model. Okay. So there are model compression techniques such as quantization, pruning, knowledge distillation, etc. Or you can use smaller models. That means you build a model, you compress the model, right? Using any of these techniques. And when you do these techniques, please don't assume that accuracy will drop significantly. Not at all. There might be slight drop. That's okay. Because if speed is more important, yeah, so be it. And Edge AI keeps data on device from privacy. So what happens here is, even in this Northwestern Railways example that I've given, human faces, we are not saving anywhere. When the data is processed on the edge, only the ultimate results are saved. The actual data is not saved until unless you want to save it. Okay, so this is the beauty. Okay. Even with respect to training, hey, what if I don't have data? I need more data to retrain the model. Sure. You can retrain the model locally and send only the model updates, which are called as gradients. Only those can be sent to the central server. So your model also is improving with the knowledge of the new data. And you're also ensuring that your raw data is always on the device and you're never using that. Apple has Siri, Google has something called as Gboard. Privacy and population learning, they are no longer mutual, right? And whenever you want to work on any AI solutions, follow one of the frameworks. It could be ML Ops, it could be CRISP MLQ, it could be Gen AI Ops, it could be Agentic AI Ops, some framework which is going to guide you step by step. Okay. So CRISP MLQ is a common shade language which can be used. And in medical AI, 80% of failures come from miscommunication, not technical challenges. Okay. So CRISP MLQ, does it emphasize on too much of documentation? Not really, right? So within each of CRISP MLQ has 6 phases, and within each of the phase, in each phase, um, you will have something called a sprint. That means all the phases you can revisit again and again, again and again, right? And at every point of time, you'll have quality gates or checkpoints. They ensure that you don't fall into trouble. Okay. So even if you want to restart from phase two, it's pretty simple because all these phases you will go through quickly. Okay. What makes ML quality assurance different, right? In machine learning, it is probabilistic, meaning it may happen or may not happen. The prediction might be true, may not be true. Okay, the prediction might be right or wrong. It is probabilistic. Whereas in typical software, it is deterministic. That means with 100% certainty, you know what the output is going to be. That's the difference. And when you do a prediction that patient might get sepsis attack, to check whether that is right or wrong, you have to probably do additional tests, or you need to give some treatment. Okay, so there is no way to check in most cases whether the predictions are right or wrong. You don't even have a mechanism to uh, and for each and everything, if you have to manually do it, then why do you need AI predictions? So that's a tricky part. And one more thing is, like software, if you try opening a specific application, it won't open, it will throw error. That's for software. But for models, they will give you some predictions. They'll not tell you that I cannot open, or such errors you typically won't encounter. That means, what if the quality or accuracy is dropping? If the quality or accuracy is degrading or dropping, then what? Okay. We also have emergent behavior, unexpected feature interaction, so on and so forth. Yeah. Next. 90% of medical AI projects fail in phase one or two, which is business and data understanding and pre-processing. Not because of data science issues, because of wrong problem definition. If you are building a model which says this model is 99% accurate for arrhythmia detector, right? And it becomes absolutely worthless if clinicians already have 98% accuracy. For that 1% improvement, do you want to do this? And what is the value of that 1% improvement? What is it helping you solve? So instead of can I use AI, your question should be should I use AI? Is AI even needed or am I force fitting? Okay. And then you need to define the success criteria also. Success criteria not from one single person standpoint. Success criteria should be defined for all the different stakeholders. You might have regulators whom you might want to satisfy. You might have administrators, you might have patients, you might have clinicians. You need to satisfy the needs of all these different stakeholders, which is very, very important, right? And another challenge that you'll face in this phase is data. You might get data, but is the data even valuable or not? Let me ask this question. For this Garba solution, okay, we want to classify or we wanted to predict whether the embryo is of good quality or not. For this, we first of all need to get embryo images. Okay, we need to get embryo images and we need to then grade and determine whether embryo is of good quality or not. Now the question mark is if these embryos are graded by an embryologist who has less than 5 years experience, can I trust? Can I trust that that person is doing correct job? That person is 100% correct in grading the embryos? Or should I rely on senior embryologist who has more than 10 years experience? Whose grading should I really focus upon? Very important, isn't it? So, first of all, data sets are rare. Even if you get, you need to assess the quality of that. So there are concepts such as transfer learning, augmentation, synthetic data generation, simpler models, federated learning, so on and so forth. Okay, next. Once you are certain that the data is of good quality, yeah, experience is one thing, Selva, and there are a lot of techniques, right? If you use devices to capture the data, then there's a technique called as gauge RNR, GAGE, repeatability and reproducibility. However, if you want to know whether the grading being done by junior embryologists is also proper or not, then there are techniques such as AAA, attribute agreement analysis, okay, where you assess the quality of different embryologists, senior embryologists versus junior embryologists. You do all those comparisons and you get some kappa values, flake kappa statistics. And based on that, you can determine the quality. Then comes model building, right? So you first of all divide your data into training data and test data, okay, which is a simple process. And then you train your model on the training data and then test it on the test data. Suppose you have a variable called um, gender, for example, okay? And say you have an output variable which is called as, let me quickly do this for you guys so that there'll be some holistic understanding. Suppose in the data set, you have an output variable, okay, which is whether people will get sepsis attack or no sepsis attack. And say you have gender and various other demographic details. Now, first, you look at the output variable. If you have, say, 10,000 data points, out of which 9,000 are no sepsis attack and 1,000 are sepsis attack. If you have this kind of data set, then your model will learn more of these patterns, no sepsis attack, right? More of these patterns will be learned and less of these patterns. Why? Because machine learning algorithm is like a kid only. If you teach more of 5 table and less of 6 table calculations to your kid, then if your kid takes the main exam, 5 table questions, they'll answer correctly. 6 table questions, because they practiced less, they didn't learn it well, maybe. So they will go wrong. If you ask a question, what is 6 by 6? They might say 30 because they are getting influenced by 5 table. So you need to balance the data. Okay. And when you build a model, when you evaluate it at the end of the day, you need to ensure that your model gives you more or less the same accuracy for both these classes. If you have two class problem, if your model overall is giving 99% accuracy, then 98%, 99.5%, whatever, more or less the accuracies of prediction for both sepsis attack and no sepsis attack should be more or less the same. And you also need to look at this gender. You have to segregate male data, you have to segregate female data. And for male data also, you do different predictions. For female data also, look at different predictions such as sepsis attack, no sepsis attack, sepsis attack, no sepsis attack. In all these scenarios, it should more or less give the same accuracies. You cannot say that, okay, in this male scenario, it's giving 99% accuracy. In female scenario, it is giving 92% accuracy. This is not acceptable. So such deep thinking when you build your models are needed. Not a lot of people do it, but when you go for government regulatory approvals, etc., all these things will be tested, especially European Union, CE marking, US FDA, etc. You will certainly be questioned on these lines. Okay. There is something called as area under the curve, this receiver operating characteristic curve. This is one way of determining the accuracy. However, as I've said, you need to look at balance versus imbalance in the data. And you also need to look at uh, predictions for each of the different categories, right? Then, if model performs worse on some demographics, like for male it's doing a good job, for female it's not doing a good job, whenever such things happen, then your model is not worthy. Okay, your model is not worthy to be deployed in production. Okay. Deployment and monitoring. Deployment means I deployed the model, job done. No, it doesn't work that way. After you deploy the model, continuously you need to monitor the model. Continuously you need to update the model. So there is something called as, okay, you might say that, do you know what, each time I retrain the model, do I need to get the government approval? Let's see. There's something called as predetermined change control plan. FDA approves model plus change protocol. What changes are you anticipating? How are you going to validate? Okay, so on and so forth. And adaptive algorithms, you need not resubmit. If you build a complete machine learning operations, build a pipeline and say that this is how the data will come, this is how it will get retrained, and this is how the model gets updated. In such scenarios, frequent resubmissions are not needed. Okay, that's it. Then, how detect deployed model failing without real-time ground truths, right? So there is something called as proxy monitoring, because you might say that my model is predicting, but I don't know the ground truth. So rather than focusing on the final output, you can focus on the inputs, right? Earlier, maybe the age group of people were 20 to 25. Now the age group of people probably have become 35 to 40. So the data itself changed. That means your output will change. Prediction distribution. Earlier, only 10% of people were being tested positive for brain stroke. Today it is 50%. Something changed, right? And then you can also bring in human into the loop, so that when you bring human into the loop, the critical cases are understood by the human. Finally, data privacy and regulatory, very, very important. You have CDSU in India, you have DPDP Act in India, so on and so forth, right? So 87% of Americans uniquely identifiable are uniquely identifiable using the zip code, gender, birth date, etc. Right? Um, and medical records contain more details. So anonymizing people is very, very difficult, because in US, you know exactly where you sit, zip number, etc. Everything will be perfect. Based on just that, you can identify who that person is. Okay. So even if you do anonymization, you're just trying to make re-identification harder, but not impossible. So in medical, you cannot say that I'll create synthetic data, artificial data, right? Even if you get artificial data, unknown biases might creep in. We really don't know about that, right? Some rare events might come in, undersamples rare diseases, right? Some unknown unknowns can come in, right? You might be, um, sidetracked by your own personal judgment, so on and so forth. So you should be very, very careful. So, how handle when AI discovers patterns contradicting the clinical practice guidelines? So, AI, first of all, will find the actionable patterns, which can also be spurious correlations, right? But when contradicting CPG, clinical practice guidelines, you have to check whether it is defaulting with the guidelines. You need to, of course, investigate rigorously. Don't just base the decisions on your thought process. Confounding variables you need to check, so on and so forth. Then you have regulatory strategy, right? For example, EU AI Act says that high risk AI, I mean, all the medical devices for that case are high risk AI devices, and double approval is needed with MDR, medical device regulatory, right? Um, so there are risk management systems, there are quality management systems, etc., 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 which are all linked to this European Union AI Act. So following a few of these standards will certainly help you fall in line. Okay, and then if you get these certifications, that in itself will move you closer to abiding by European Union AI Act. Okay. In India, we have CDSU. That's that is new to AI regulation. They're bringing in those aspects now. So you learn from FDA, EU mistakes because they are coming up with new regulations now. But the disadvantages are that um, since it's new, requirements may frequently change. So frequently you might have to redo things. And whenever you want to get an approval of your uh, software in medical device, these will be called as software as medical devices, software in medical devices if it's edge-based deployment. Ensure that you always go with the toughest of the standards so that in India it becomes easier to get the approval. Fine. So this is all I had for the day. Hopefully, I could give you some meaningful value. Um, but greatest risk is not failure, right? It's subtle failure. If the clinicians are going to trust the machines, they don't understand. And if they're going to make the decisions on top of that, that means it's going to be very, very dangerous affair, right? And this is what, let me see if I can show. Yeah, this is what we have also experienced. When we deployed this solution, in 7 clinics, it is being used. In a few of the cases, we started tracking the number of cases where they were spending time to manually grade embryologists before making AI also give the predictions. And over a period of time, they started over relying on AI. And earlier, they used to take close to 15 minutes on each embryo to grade. We started tracking those metrics. And after we gave this AI solution, they started spending hardly 5-6 seconds. That means they overly started relying on AI, which is a risk. What if AI is continuously predicting incorrectly and continuously highlighting wrong portions? Then these embryologists also will start believing that that is true. So this human AI is very, very important. Human AI collaboration is extremely important, right? In certain cases, remove AI support, don't give them. In certain cases, randomly say that some 10% of what you have graded will directly go to the senior embryologist. Whether the AI prediction will come or not, that person doesn't know. So that is how you have to build your system so that they now ensure that they're completely focused on what they are doing. Okay. In 10 years, any guesses, friends, on what percentage of diagnosis will involve AI? How should medical education change? Any thoughts? Any closing remarks? Any closing remarks, anyone? What is your thought? In 10 years, what percentage of diagnosis will involve AI? 80 to 89%, sorry, 80 to 90% by 2035. It will involve, not replace. Okay. AI is just an assistant, especially in the space of medical. Here also you can create different workflows. Very simple workflow, automate it. Complex workflow, human in the loop. That's it. Ethics, the communication, empathy aspect, AI cannot replace. You have to depend on humans. And human AI collaboration or teaming is the way to go. Okay. What is the one thing you will do differently in your next medical AI project? Right? If I were to ask you that, obviously, you have to involve clinicians at the start of the project. You need more data quality time. You need more data, you need more quality, and you need to spend more time on acquiring quality data. And you have to monitor the solution right from day one. Okay, clinicians must ask hard questions, right? And also, they have to demand certain performance metrics. And as leaders, you have to provide some money, support budget for post-deployment monitoring. Please use cross-functional team, engineers, clinicians, make them work together. And such workshops, right? It's not what you learned, it's about what you learned differently, right? So that makes a difference, certainly. And the goal of AI in healthcare is not to make machines that think like doctors, but to help doctors think like the best versions of themselves. All right. With this conversation, I would like to stop here. Thank you so much, Sujata ma'am, for providing this opportunity. Hopefully, I gave you some meaningful insight. Thank you and all for your futures.
Yes, sir. Thank you. Dear participants, if you have any questions, please post in the chat box. Otherwise, you can unmute yourself. You can directly contact the speaker. There are many positive responses from the participants. No questions are posted. So there is a question, how the AI and signal processing are related? Sir, I think you are you have to unmute yourself, sir. So your voice is not audible.
Yeah. So to build AI solutions, you need data, and you get a lot of data from signals. And we have to do the pre-processing of this data that you're getting from signals and make it good quality data and supply it to your machine learning algorithms, which will ultimately help you build good AI solution, Dr. Ravi, right? Then Silva Kumar has a question, I think. Is it possible to create a smart environment to monitor the growth of embryo? I didn't quite understand what you mean by that, but if your AI or machine learning model goes inside the microscope, then I think that's the gold standard. That means that's the the best possibility. Okay. All right, man. Thank you so much. And thank you for having me here.
Yes, sir. On behalf of AICTE Atal Academy and SNIST, I extend my sincere thanks to you, sir, for delivering such an insightful, enriching session on AI for biomedical signal processing.
UG level,
Uh, Sir, sorry, sir. Someone is asking question.
I think by mistake, Dr. Avi is unmuted.
Okay. Okay. Sir, your clear explanation of complex biomedical signals coupled with practical insights into AI-driven feature extraction techniques has greatly enhanced our understanding of how the systems are transforming modern healthcare diagnostics. The way you connected theory with real-time applications made the session highly engaging and valuable for all the participants. The session has inspired our participants to explore advanced research and applications in biomedical signal processing. Once again, thank you, sir, for your valuable contribution to this FDP. We look forward to your continued association with SNAST in the future, sir. Thank you, sir. Thank you very much.
Thank you, man. Bye.
Bye, sir. Participants, please stay back. We will start our session in next 2-3 minutes. Once again, good evening, the participants. Welcome back to the session 2. Now it's my privilege to welcome Dr. B. Suresh Kumar, a distinguished academician, industry professional with over 12 years of experience spanning higher education and the IT industry. He is currently working as a senior software engineer at XLR Software Private Limited, Hyderabad, specializing in Salesforce, generative AI, and enterprise automation solutions. Dr. Suresh Kumar holds a PhD in computer science and engineering from JNTU, Hyderabad, and has served as an assistant professor for several years. He is an active researcher in deep learning, computer vision, and human activity recognition, with more than 20 international journal and conference publications, including reputed platforms such as IEEE, Springer, IOP, and Imaging Science Journal. His strong blend of research expertise and hands-on industry experience makes his sessions highly insightful and application-oriented. So we are delighted to have him to share his expertise with us today. I now hand over the session to Dr. Suresh sir. Sir, kindly proceed with the session. Sir, you are not audible. I think you have to unmute yourself.
What about now?
Yes, sir.
Yeah. Thank you very much, ma'am. So today, good evening, everyone. Hope I'm visible. Let me share my screen. Yeah, I hope my screen is visible.
Yes, sir.
Yeah, thank you. Thanks for the confirmation, madam. Okay. So, as today's session is on, this particular session is on the hands-on implementation of deep learning for the medical image classification. So the thing is, why do we need to work on this, particularly, in the field of medical images, and why do we need to implement this particular medical imaging for the, using the deep learning kind of things? And basically, what are the different types of things are there for the medical images? And this is not only the theory session, and we are going to have a hands-on session with some practical knowledge. I'm going to explain, how we are going to use the CNN for the medical images and all those things. Okay, so I'm going to turn off my video. It's like I'm watching myself. That's the reason I'm turning. Okay. So basically, if you see, today, medical imaging is the heart of the modern diagnosis. Every hospital generates a massive volume of X-rays, MRI scans, and the CT scans every day. In large hospitals, like, doctors have examined the hundreds and even thousands of images daily. So this is not only the process of time consuming, it is mentally, exhausting, the doctors for, to do the diagnosis and all those things. And there may be situations where the two or more doctors sometimes interpret the same image with a slightly differently. They are going to analyzing and they are giving the different, precautions for the people. So, and during the situations like pandemic or the mass screening, it becomes practically impossible to manually analyze everything with a several, with the same level of attention. So this is where the deep learning comes into the picture. So deep learning models do not relay or manually write the rules. They learn directly from the data. They can look at the millions of images automatically and learn what type of patterns we are getting, and that is called, we are calling it as a feature learning. So, and it is the main reason why deep learning is evolved in the medical image analysis. Okay, so this is what the thing, why the deep learning is main, placing the major role for this medical image classifications. So you don't need to, to be a deep learning expert to follow this session. We'll go step by step, and maybe we'll go with some basic part to how the CNN works and how to implement the things by using the CNN. Those things we're going to see. Okay, so let's see the roadmap, how we are going to, what we are going to see in this particular thing. Before we are going to deeper, let's have a clear idea on what we are going to see in this one. Is like, I'm going to explain about medical images and using how to do the data pre-processing, data sets, from where we need to get the data pre-processing, and a deep learning for the images. Okay, so think of this as not a lecture, but it's like a lab session where we'll write the code and run it, understand every stage. Once you understand this pipeline, you can easily extend this to the MRI, CT scans, multiple class problems, or even your own ideas. Okay, so this is a basically we are going to follow the procedure. Like we are going to introduce the medical imaging, and we are going with the data sets, like different types of data sets available in the Kaggle, or freely available data is available. Like, okay, and we're going to analyze those data with a deep learning kind of things. Then we're going to build our model, basic model, CNN model. Okay, then we'll evaluate and test it. What are the limitations and the best practices we need to follow? So those things we're going to see in this particular session. Okay, let's see what basically what is the medical image classification. Let me just basically. Right. Let's first clearly understand what is the medical image classification means. In simple term, medical classification is the task of taking the medical images such as the X-rays, MRIs, CT scans, and automatically assigning the category or the disease of that particular class using our AI system. For example, we gave a chest X-ray to the system, and it tells whether it is a normal one, or the, like, let's say any pneumonia kind of, like, let's we are working as a session, practical session, we're working on the pneumonia X-rays. Okay, so that is the reason I kept the pneumonia here. So for example, like I'm saying, like, where if we give a image to the system is going to, our deep learning methodology system, is AI system, is going to provide is that the normal or the pneumonia X-ray. So the simple idea here is applies to many medical problems, like a brain MRI can classify it's tumor or not tumor. So it can be classified like tumor or not tumor. And one more thing, like a retinal image can be classified as a diabetic retinopathy normal image. So like that, we have a different types of images are there, so where you can work with the medical image classifications. So conceptually, the problem is very simple. We give the medical image to the computer. The computer analyzes it using the trained deep learning model, and it produces the prediction. But what happens inside that model is extremely sophisticated. So the model is not looking at the image like a human. It's looking at the numbers, patterns, and the statistical structure. So that is where our deep learning works here. So if the problem is very easy, we could solve it very simple by using the simple ways. But the medical images are highly complex. The same disease look different for the different patients, right? So that was the reason the same X-ray looks different depending on the machine, angle, stage, disease. Like, there are multiple things which are impacting the our images, X-ray or MRI or the CT. Even expert doctors sometimes disagrees that so is that that person is affected with that particular disease or not. That tells us that is like it's not a simple pattern matching problem. So earlier approaches, like where defining the features, edges, shapes, into, like there are multiple things are there where machine learning models are worked, but someone had to decide what feature is the matters, correct? So and in medicines, the extreme difficult one is because of the disease do not follow the same geometric rules. Let's say if you are familiar with the machine learning kind of things there, if you remember, if you have any data set like house price prediction data, consider the house price prediction data. In the house price prediction data, you have a same kind of things, like you have a what is the size of the house, how many bedrooms are there in the house, how many washrooms are there in the house. So there is the same set of patterns will be there. But coming to the medical images, it is quite different, where you're going to see the different patterns in the images. So that is where the deep learning changes these approaches completely. Instead of telling the computer what features look for, it gives the large number of labeled examples. So where itself it is going to learn the things, which is, we can call it as a feature learning. So why this is powerful? The model can discover the patterns that human cannot explicitly describes that. That was the reason the deep learning is placing the having the major role in this medical image classification. So from now, whenever you heard you hear about the medical image classification, think of it as a function where you are giving the you are giving the image into the intelligence system. The intelligence system is going to give you the medical decision output, like the person is affected with some let's say pneumonia or not, or tumor is there or not. So like that, it is going to provide you the functionality. So why is medical image important in medical image classification important in healthcare? So there is few things. The key reasons here is it is used to early detection of the critical diseases like pneumonia, cancer, tumor, strokes, and faster screening. So which is going to gives the immediate results. Let us understand why this problem is so important in the healthcare. Today, hospitals generate the massive number of medical images every day, like X-ray, CT scans, and all those things. But the number of trained specialists who can analyze that images are limited. So this creates the huge workload on the doctors, maybe the radiologist or the doctor, whoever it is. So in many medical conditions, time is everything. If pneumonia, like if cancer or pneumonia or stroke or any internal bleeding like detected early, the patient's survival chances improves dramatically, right? So that is where the important, this image classification is very important. Even the best doctors are the humans. They get tired. They work under the pressures. They have this, they have to scan hundreds of images daily. So this is not because of the care, this is not because of the things we don't have, like the system, AI system is not going to fit. Yes, AI system is going to fit into that. But the problem is we have the less number of doctors who are analyzing that particular thing. AI does not replace the doctors. AI acts like a very fast and very consistent assistant. It's like an assistant. It won't give you the final result. It just assists you to, is that person is affected with that particular disease or not. So then, what are the challenges? Like, what are the challenges are there in this? If at all, if you apply, medical image classifications using the deep learning and all those things, medical images, challenges with the medical images, right? So, like, if you see, we have X-ray images, we have the MRI images, right? So let's talk about the first and mostly widely used medical image technique, the X-ray map, X-ray imaging. So X-ray have used in the medicine for more than a century. They are fast, cheap, and available almost every day, everywhere. So here, and by taking that X-rays, this if you give it to the system has to analyze that, right? So the main challenges here is high variability across the patients and the machines. Where the machines are less, the patients are high. Even with a CT scanning, MRI scanning, whatever it is. Okay, so that is where, so far, like, everything may sounds very promising, but now we must talk about the hard part of here. The medical images analysis is not an easy job, right? In fact, it is one of the most challenging area in AI. There are several serious problems that makes the task difficult. So first medical images are often noisy, blurred, and the low contrast. So this can be happen due to the patient movement, limitations of the image devices, and all those things, right? So the second one is the disease can look differently for the different persons. So these are all the different types of challenges are there in the medical images. So given all these challenges, you might ask, like, why use AI at all? The answer here is deep learning is currently the best tool we have to deal with this level of complexity. Okay, so like, let's say now we understood the challenges. And next, a logical simple question: why AI, and especially deep learning? Still the right approaches for, is this the still right approach for the medical imaging? So let us answer this. So why AI for the medical imaging? So after seeing the challenges in the medical images, like, as I said, there is a blur, the movement of the person, and there is no clarity in the images at that time. So at that time, why should we even use the AI for this problem? Off the network. Hello. Yeah. So the thing here is, okay, so why the machine learning, why AI for the machine learning, for this medical imaging? So just now we have seen what are the different types of challenges are there in the medical images, like medical imagings. Like, so because of the the person movement, because of there is a blur in the images. So because of all those things, there may be a chance he may person may get the different diagnosis. Because say, let's say the person has got the pneumonia. Maybe when he's the person whoever is taking the x-ray, there may be a chance of photo problem. There may be a the moment of he may move here and there, the person. So at that time, it is very difficult to take those take the pictures of those things, right? And one more thing is, earlier people are trying to build the rule-based systems. Like, say, if if this type of shape appears, if there is a round circle appears, then this is that is a particular one type of disease. Then what is a normal one, what is the intense one? So, but medical images do not follow the simple rules. The same disease can appear in the many different ways. So the systems were the failed in the real world settings. So here we are going to work with the deep learning. Okay, so we are going to work with the deep learning, which is we can call it as a feature learning. And in this, especially we are going with the CNN networks. So you can ask me, like, why CNN? So CNN networks are designed to work with us, data like the images. They learn the local patterns, combine them into larger patterns, and finally into the high level of concepts. So this is exactly how the medical abdominal appears in the images. So once trained, AI model can be analyzed as thousands of images in a minute, correct? So you have, let's say you worked on a one particular data set. You can save that model. Later point of time, you give one more image to that. So you build a model and you kept that model aside. I mean, you saved that model and you can use that model for the remaining images. So in that case, you are reducing the lot of human intervention here, right? So the important thing here is, however, it is very important to say this clearly, AI is not the replacement of doctor. I'm saying it is a decision support system, right? So maybe whenever we enter into this deep learning kind of things, right? So maybe you can you may think like, okay, so deep learning maybe replace the doctor, or AI replace the doctor. No, just it's like a supporting system, right? Now let's understand how the medical image classification pipeline, what we need to do. Okay, so here in this process, what we will do is, now that we understood why AI is used in the image medical imaging, like now let's look at how the medical imaging classification system is actually built. No matter whether you are working on the X-rays, MRIs, CT, or any kind of images, the overall pipeline is almost the same. You can take anything. If you want to work with, X-rays, you can. MRI, you can. So, but the process is same. So first thing is the data collection and the labeling. So everything starts with the data. Everything starts with the data. That's same thing we can call it as an image acquisition. So we collect the data, medical images, whether directly from the hospitals or the publicly available data set. You can go with a Kaggle, there you will find a lot of data sets. So these images come from the scanners, are stored in the digital files without good data, or no AI system can work. Right? Without good data, there is no system, no AI system can work. If at all, if you have the good data, then only the system will work perfectly. So that was the reason, whenever you are working with any data, choose the data correctly. And the next one is the pre-processing thing. So the raw medical images cannot directly fed into the neural network. We usually resize them, normalize the pixel value. Sometimes we have to remove the noisy and enhance the images from the system, contrast images from that particular data set. So this particular steps ensures that all the images are having the consistent and the format for the training. The first step is always the data collection. You need to collect from the Kaggle or UCA machinery data set. Like there are a lot of websites are there where you can directly get the data. Right? So once you get the data, the important step is the pre-processing. Most of our work is goes on this pre-processing and the data augmentation part. Sorry. Okay, so this part. Data processing is the important one, because if we train the system with the unnecessary data or the noisy data, then system will remember that data and will store the features of that data, and maybe there may be a chance it may give the wrong predictions also, right? So that was the reason always we need to go with the pre-processing. In the pre-processing step, as I said, we need to resize all the images. If you are working with the images, you need to resize the images, right? So because you cannot have different size of images. You need to have a clear cut of images, like how they're working and how they are stored, and how we are going to processing that type of images. And the next one is a model design. So here for the model design, we are taking the simple one, that is a CNN. maybe you may have the LSTM, RNN, GN. So there are granular neural networks, long short-term memory, and RK, recurrence neural networks. All those are there, but basically for our understanding, we are taking the CNN, which is a simple one to understand, and so it's like a heart of the system. The main in the main classification problem, this is the heart of our system. So the heart of the system is the CNN, convolution neural network. So this is the component that learns to extract the features automatically from the images. It starts with the simple patterns and gradually learn more from the complex structures. Okay, so where it is going to, when we give the input, when you give your input data to the system, and before that you need to do some, pre-processing and the data augmentation steps and all those things. And once that is completed, then you need to proceed to the CNN. I'll show you how the CNN works. It's very simple to understand and all those things. So during the next, once the you provide input to the system, then it will move to the training phase. So during the training phase, we give the model both the image and their correct labels. So for that one, right? So normally in supervised learning algorithms we have, right? So where supervised learning algorithm, we are going with a we have different types of algorithm. Okay, so where the supervised learning algorithm basically works with the label data. So those kind of things we will apply here. So the model makes a prediction and compares them with the true labels. And let's say if anything goes wrong, again it adjusts the internal parameters. So this process is repeatedly done many times until the model learns the useful patterns. So once the model is trained, then what is the next step? Then we need to go with the evaluation step. So we must evaluate the data it has never seen before, the extremely important one. We want to know whether the model has really learned the general patterns or just memorized the training data. So now, when the final image comes, we pass it through the trained model and get the prediction. This is what happened in the real cleaning system. New image in, the prediction output we'll get the prediction output. So there are the two phases here. One is the training and the inference. So training is slow and computational is heavy here. Inference is fast and what we use in the real world applications. Correct? So that is what we can, that is what we need to do for this CNN and implementation purpose in the classification. So in today's hands-on session, what we'll do, we're going to implement every block of these pipelines ourselves, so like loading the data, pre-processing, and we're going to building the CNN, and we're going to training it and evaluating it on the data. So now, I hope that you have understood the pipeline. If you go to any type, if you take any other images, like if you go with the X-ray or MRI or any type of images, so this is the same process we need to follow. Okay. So why CNN? And what is the convolution neural network? So basically, yeah. Right. So coming to this, what is the convolution network and why we need to apply this only CNN to the system and all those things. Now we come to the most important technical concept that of our today's session. So convolution neural network or the CNN. Almost all the modern medical images AI systems are based on the CNN. So we try to use a normal fully connected neural network for the images. We immediately face the huge problem. Let's say we have a 224 by 224 images, which has the more than 50,000 pixels. connecting each pixel to each neuron creates the millions of parameters, which is the insufficient, right? Which is going to leads to the several outfitting of our data. So more importantly, images are not just the random numbers, right? So where it is going to connect it to the nearby pixels. Patterns like edges, textures, and the shapes and the locals. So the fully connected network ignores the structure. So CNN are specially designed for the exploit the spatial structures in the images. So CNN works by looking at the small local regions of the images by learning the simple patterns. Then it combines the simple patterns into one, more complex ones. This happens by layer. If you see, we have the different types of layers here. So for the medical abnormalities like are not defined in the one pixel. They are defined by pattern, right? Pattern of textures, shapes, and the structures. So CNNs are extremely good for this capturing these kind of information. The most important is different from the classical methods is we don't tell the CNN what features you need to look for. We just give the data and it will learn the features itself. So this is why the deep learning works as well as in the complex domains like medical imaging. Okay. Let's say in the research perspective, like almost all the state of our medical image systems, okay, are based on the CNN variants only. Okay, so let's see the architecture of CNN. Okay, so this is the architecture view of CNN where, okay, so now we understood like why CNN. Let's see the architecture. So how the network is constructed. So here the CNN has several types of layers where layers arranged in the specific order to perform the raw images data into the meaningful predictions. So let's say here I'm giving the input. This input has to go through the different types of stages, and finally it will store you into the one layer. Let's say here we have it starts with the input always. It starts with always the input. So here inputs is nothing but which receives the input data. For example, chest X-ray. This might be the 224 by 224 grayscale image or another any standard size. So the input does not learn, but it defines the shape of the shape, how the flow, shape of the data that will flow through the network. So the second thing is the convolution layer. Second one is we have the convolution layers here. These are all the parts of the layers, convolution layers here. So the convolution layers are core building blocks of CNN. These layers applies the learnable filters that slides across the image to detect the patterns like edges, textures, and the shapes. So each filter produces a feature map, which is going to represent the which is going to representation of where the certain patterns occur. Stacking multiple convolution layer enables the network to learn abstract features here. Each layer, in this case, detects the simple edges and the deeper layers, which is going to capture the shape of that particular decision. This is what is going to happen. And if you see, there are some, max pullings and fully connected and all those things are there. There are certain things like activation layer, pooling layers. Okay, so like after each convolution layer, right? So we apply the non-linear activation function, mostly like ReLU layer is there, and ReLU unit will apply. Activation function will apply. So basically, ReLU introduces the non-linearity, which is allows the network to learn the non-linear combinations of the features, something like the something crucial for the complex pattern detections. And if you see here, we have the pooling layers. Don't worry, I'll show you how the CNN works. So we have the pooling layer. Basically, we have the max pooling, mean pooling. Okay, so like that, we have different types of pooling layers, which follows the convolution layers to reduce the dimension of our features. So mostly, commonly, we are using the max pooling, which takes the maximum value from the local pattern. Okay, so once that is completed, right? So we'll be moving to the next one is the fully connected layer. once the convolution and the pooling layers are completed, right? So the stage have been which is extracted the features. Then we need to flatten the features, all that features. So that can be feed into the that can be feed into the fully connected layers. So these layers acts like the traditional neural networks, where they can take the learned patterns and make the actual classification decisions. So usually the final layer uses the activation like softmax, sigmoid output class probabilities. So the strength of this CNN lies in the hierarchical flow here. The first one, if you see, we have the convolution layer. So which is the feature extraction with the convolution and the pooling followed by the fully connected layer, right? So the earlier layers captures the features and the patterns. The deeper layers captures the high level of semantical meaningful features. So that was the reason we are saying like hierarchical learning, why the CNN is excel in the image tasking. Okay, so that is what will be happen in the thing. So let's understand how, okay, before going to this data set and all those things, how the data set is structured and all those things, let's understand how the CNN works here. How the deep learning, how the CNN works in the deep learning. So these images I have taken from Google, okay, so which is better for understanding purpose. That is the reason I have taken this. Let's say, okay, so sorry. Okay. So let's consider this is an image. This is an image. So whenever you send this image to the system, how our computers read this images is, let's say this image with the size of like by BA by 3, that means the 3 depth is red, green, blue, right? So the image will be stored in the form of the matrix, which is the pixel values. This is how the system will see this particular image. When you send your system to the, whenever you send the image to the system won't see it as an image. It will see it as a matrix. Simply, when I simply saying it will see it as a matrix. So once that it is done, right? As I said, it will move to the convolution layers. It will move into the convolution layer. Let's see how the convolution layer works. As I said, firstly, it will go to the convolutions, ReLU layer, poolings, and the fully connected layers. Let us understand. Here we have the image like X, image like O. So whenever we provide this particular image to the CNN, the CNN has to get to where the pattern is available. See here, it is in the X format. It is in the O format. The symbol, O symbol, and it is in the X symbol, right? In this case, whenever you're sending your image to the CNN has to get only this data, not the remaining data. Right? So how it will work? See, this is the case here. So there may, whenever you want to extract this, there may be different images you may get. Correct? It may be sometimes it may be like this, sometimes it may be like this, sometimes it may be like this. But all those things are belongs to the X images only. The similar way, image, when we go to the medical image diagnosis also, you may get a different types of images, like, right? So, but finally, you need to get the clear idea about that. So as I said, it's the X image, this is also X image, this is also X image, and it is also O image, right? So let's take one image and get the details. Now, so here, how the CNN works here is, it is going to consider like this X is equals to this X, this O is equals to this O. That means wherever you see this type of features, it has to get the details. It has to extract the details. Now, for example, we are going to keep the black pixel values with 1, but wherever the black is there, we're going to keep the 1, and the remaining things, we're going to keep it as a -1. Wherever the data is not available, we're going to keep it as a -1. So I'm considering the -1 wherever the data is not available. Wherever the data is available, I'm considering it as 1. So this is the first step. Now, using the normal techniques, when we compare these two images, let's say I'm comparing these two images here. One is the exact image, second one is this image. Let's compare these two here. This particular portion is not available here, and this particular portion is available here. I'm combining these two. When you combine this, wherever the possibility is there, let's say, okay, let me put, just a second, pointer option, pen. Okay, see here, this option, this particular point is not available here. When I'm combining these two, it is going to be come here. Okay. Next one, this option is available, but this option is not available here. I mean, this one is not available here, but it is available here. And this value, this portion is available here, but it is not available. But as I'm combining the pictures, so I'm having all the kinds of like these rows, I mean, these values and these values. Okay, all the values are going to be added into my final image. Right? You're going to see all the images here, all the pixel images here. Right? Now what we are going to do here is the CNN compares the images, the piece by piece. The piece look, it's called as a features here. We can call it as a features here. It's going to call it as a features. So finding the rough features of the images, wherever the same position of the two images are there, CNN gets that similarity from the images. Let's say here in this case, these two are available here. Right? These two patterns are available here. It's going to extract that. And this pattern is available here. This pattern is available here. So it is going to extract the patterns from the given system. Right? So after extracting the features, then we're going to apply the filters. Okay. So there are different types of filters are there. Okay. So Canon filters. Okay, different types of filters are there. Kernel filters, like that. Different type of filters are there. Okay. So let's take any type of filter to apply onto this particular CNN. Now here what we are taking is we are taking one simple filter, which is let's take the simple one. Okay. Now this particular filter I'm applying onto my image. So what will happen here is I'm taking this image, this particular filter, applying to my wherever I'm seeing the ones. Now see here, that's step one. Line up the features in the image and the image, multiply each image pixel by the corresponding feature pixels. That means, let's say here, this is my filter. This is my filter. Okay, this is my filter. I'm going to apply this filter on the image. So that means I'm keeping, let's take, I'm keeping this image to the this portion. Whenever you keep this particular image here, so whenever you see the one, let's say 1 and 1. 1 into 1 is going to get the 1. So real time image, we are going to keep that one here. Similarly, we'll do it for the next one. So -1 into -1, again we'll get the 1. So similarly, we'll do for all the things. So this step will be applied to all of this image, all of this entire data. Once the filtering is applied, what's the next step? We need to apply the, okay, so here we are applying the average filtering here. Let's see. So all the values we are taking, -1, -1, we have got the 1 here. So that means upon adding all these things, upon applying the filter on this particular set, I'm getting the final output as 1. So this entire field, this entire strip, in this entire strip, going to replace it with 1. That means my 1/4 this is 3/3 matrix. This 3/3 filter has converted my image into a single one-dimensional matrix. Let's consider if at all if I apply the same strip here. Okay, so into the all this thing here, then our image is going to convert it into, it is going to reduce it. Okay, so that is what the thing will happen. Let's say the same image, my same filter I'm applying, I'm sliding onto this particular portion. We are sliding onto this particular portion. In that case, again, the value will become 0.55. Right? So, by applying all these things, my image will convert it into reduced into this type of image. Right? So, where if we apply all those things, then we'll get this image. Then if we apply the remaining filters also, on this data set, I'm applying the first filter where I got this output. On this second image, I applied this image, I got this output. This is the same image, I got this filter, I got this output. Different filters we have applied, and we got the different outputs. Once this is completed, then we'll apply the ReLU layer. So what ReLU layer will do here is, it will observe each and every data. Whenever you see the negative value, it is going to replace with a 0. Whenever you see the positive value, it is going to keep that value as it is. See here, the ReLU layer will remove every negative value from the filtered images. If at all, if you find any positive value, it's going to keep the value. Let's see the functionality of this one, f of x. If it is 0, it is going to be the greater than 0. If it is x, then it's going to keep the x0. If it is x value is less than the 0, it is going to keep the 0. If the x value is greater than 0, then it is going to keep that value as it is. So with that, what will happen, if you see in the previous image, we got a lot of negative values here, right? Correct? So when we apply this ReLU layer onto this one, right? So all these values is going to be replaced with the zeros. Okay, so that is what will happen by whenever we are using this ReLU layer, see, removing the negative values from the system. It is going to help us to removing the unnecessary data. Like, what is a minus 0.11 value? If you convert this value into the color image, again you will get the black picture only. So that value will be converted into 0. Even 0 is nothing but the black image. So that is what will happen here. So similarly, we are going to convert all those things. See the values wherever the negative values are, that is going to be converted into zeros. All the negative values will be converted to zeros. Then on this, see the first image, we are applying the ReLU layer, is connected like this. Second image like this. Third image like this. Once this is completed, we are going to apply the pooling layer. The pooling layer, we are going to take the size as 3 by 3 size. Okay, so we are going to apply that pooling layer onto our window. Simply like this. So what is here? As I said, we are going to apply the max pooling. So the max pooling is going to place our we are taking the 2 by 2 strip here. So when you take the 2 by 2 strip here, when we're going to replace, when we're going to place here, let's say I'm replacing here, right? So here the 0.7010. So the max pooling, the max pooling is like, it is going to take the maximum value from that strip. In this first strip, if you see the first strip, the maximum value here is the 1. So for this entire value, it is going to consider as a 1. Now it will move and it will place the a strip here. So in this case, the maximum value is 0.33. Again, it's going to convert into 0.33 here. Like that, the strip will move and it's going to replace those values with maximum values. Right? So once this is applied, right, our image has been, the image has been converted into like this. We have 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. So 7 by 7 image. 7 by 7 image has been converted into the 4 by 4 image. I mean 4 by 4 matrix. 7 by 7 has been converted into the 4 by 4. Right? So this is what will happen in the CNN. So because it is extracting the features, important features. If you see the first one, just normal filters has applied and is converted the it is taken the average value and is replaced that. Then after again we are selecting, we are reducing the features. We are not the features, like we are reducing the pixel values to get the accurate features. So this is what happened in the max pooling layer. So once the pooling layer is completed, so all the strips will be converted like this. Then after this is my original image. On this original image, first initially we have applied the convolution layer, which is applying the filter. We are applying the filters and all those things. Then after we have applied the layer. Then after we have applied the pooling layer. So this is a one step. It's the initial step. So once that is the first step is completed, then we got output like this. So this is by 4x4 matrix, the 4x4 matrix, and here we have the 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9x9 matrix has been converted into the 4x4 matrix. That means features we have extracted here. So from this entire image, which is having the negative and the positive values, we have done the convolution layer, we have applied the ReLU layer, we have applied the pooling layer. After that, we have got a feature without any negative values here. Then what's the next procedure? Then we are going to apply this one to to extract the number of features. If you want, to extract the number of features, again you can go with a 2D layer or 3D layer, like that you can do. So then I'm applying the one more repetition here. So after one more repetition, I got the 2x2 matrix finally. So this 2x2 matrix will be given to the fully connected layer. So whenever you're sending the data to the fully connected layer, you need to send it into the form of the strips here, single list. So whatever the 2x2 matrix are there, that will be converted into the single list format here. Okay, so this is for the single list format. So we are doing this for the x value. Only for one x value, we have done all these things. I mean, what's x image, right? So this process will continue for all the images, for 0 also. Whenever we done for the x value, we got this string. Whenever we can call this a vector, not the string, we can call this a vector. So we got this vector where it's going to store in the image. The second one is we got this image where it is showing you the it is going to showing you the 0, over here, right? So in this case, whenever you want to supply the next input to the system, that input has to go through this procedure. Whatever the procedure we have done now, it has to go through that particular procedure, right? Because once that is that comes with a convolution and pooling layer, ReLU layer, and all those steps, then that is also converted into the vector. The final output is going to map with this particular thing. Let's say if you give any next input, right, by comparing the input vector with the x, let's say if the input is input image I'm giving here, this is my input image. So when we can get this, once we complete the convolution and ReLU layer and the pooling layer and all those steps, it is going to convert it into the vector. That vector we are going to we're going to map with our vector x. This is the input image vector. This is the vector image x. In this case, the values have been summarized here. We'll get the sum of this one, which is going to come, which is going to map with our values. So this is going to be the 0.91% is mapping. If you combine with next vector 0, okay, so let's say if you combine with the next vector 0, there may be a chance we are going to get the 0.51, 0.51 here. So based on this classification, we can say like that particular image belongs to the class X. So this is what the procedure will happen in the input image when we send the input image to the system. So overall structure here is, so the input, whatever the input you have, you need to provide to the convolution layer. After that, it has to give into the, there you need to apply the filters, right? So, and one more thing is you need to decide on how many layers you are required. It may be the 32 by 32, 64 by 62, like that. So on this case of, like, we need to decide on the 32 by 32 or 64 by 64 based on our image input size. And if at all, if you want to go with a multiple convolution layer, you can go with that. So once you've done that, then you need to take the filter, what type of filter you want to apply in the convolution layer. Then after, select the ReLU layer or other any activation functions. Then go with the pooling layer. So once you got the pooling layer, then convert the image into the vector format. Then only you need to apply that to the fully connected layer, which is going to store all the features. So the next input, whatever the next, once the data has been trained, your data has been trained, then what we need to do? Then you need to apply. You need to apply the same whatever you have done for the training process. You need to do for the input images also. So the input image has to go through this process. Okay. So most of the people thinks like, okay, so we have trained our system. So directly, can I give the, can I give input? Yes, you can give, but the input, whatever the input you are giving to the system, that will go through these steps. Because system, how does the CNN or the deep learning understand that particular image is coming from this particular, PowerShell? That is what will be happening in the thing. Okay, so this is step we are, this is the steps we need to follow as part of the deep learning. Let's enter into the practical session. Okay, so let me go through these questions at the end of the session, guys. Okay, let me go through that things at the end of the session. Now, so here, what I'm doing is I'm taking a pneumonia data set. Okay, so I'll show you what is the pneumonia data set and all those things. Okay, so this is the pneumonia data set where it consisting of 5,863 images with the two different categories. One is yes or no, like the person is affected with the pneumonia or not. Okay, so that is what the images is consisting of. If you want to download, right, so just click on this option. You can directly download this data. Otherwise, just put on, you can take this particular image to connect with your Kaggle data from, to connect with the Kaggle hub, from where directly you can connect to the data set. But take care whenever you are connecting to this, whenever you are downloading or connecting to this Kaggle, so please take care, like you need to set up the API keys and all those things. But okay, so you can also set up the API keys and all those things. That's not a that's not a problem. Okay, so I'll show you, like you can directly download. Okay, you can store it into your Google Drive, and you can mount your Google Drive to the Google Colab. Okay, so I'm executing this things in the Google Colab. Okay, so which is going to have a different kind of things here. So yeah, this is about the data set where we have the 5,200 trained in training images. Like as I said, here we have the so 5,663 5,863 images, right? So this one, like if you see the just I'll maximize this. Right. So this is a normal X-ray image. This is a bacterial, formation and the viral, pneumonia, bacterial pneumonia and the viral pneumonia. So the system has to identify that this, is this a bacterial pneumonia or viral pneumonia? But only the categories are the only two categories. Like, do we have, X-ray, that image is normal or the pneumonia image? Let's see here the content. The data size is organized into folders like training, testing, and value, containing the folders like pneumonia, category as pneumonia or the normal. Right, the same thing. That's what I'm explaining here. Okay, so just if you want, you can just go through this, pneumonia data set where you can have a clear idea on it. But for me, like I'm going to this particular data set. In this data set, we have and we are taking for the 15 1,200 signing images, which is okay, so which is with 3,800 pneumonia and normal images. Okay, so and for this purpose, right, for that, to work with this CNN and all those things, we required some libraries which is going to help us to implement this process. Okay, so for this one, right, we are going with a NumPy where we need to work, as I said, right, so it's going to consider the image as a NumPy matrix. So that is the reason we are using the NumPy as our to convert the for the to store the multi-dimensional arrays like matrix. As I said, it's a image is nothing but the matrix. So to store that type of matrix, we're taking the NumPy here and the Matplotlib libraries to visualize our outputs, like what is the accuracy rate and what is the how our system has been trained, like what is the precision, recall, those kind of things. We are using this Matplotlib library for the images. To deal with our images, taking the computer vision to here, V2. Okay, so to interface with the to interact with the neural network, and that we're taking this keras and all those things. Okay, so this is what we are going to do here. Now, coming to this particular thing, as I said, we need to import the data set. So first thing is we are importing the NumPy, TensorFlows. Okay, so NumPy, TensorFlows, Matplotlib libraries. Okay, so for the data generation, like we're taking this one and to store the images and all those things. This is the normal. Okay, so normal phenomena we are for the optimizers and all those things. And here one more thing I forgot to explain about the weight optimization techniques. Okay, so in the CNN, right, so we have the feed forward and the backward backward things are there, where when in the CNN architecture, right, let me show you here. I think I have one more. Just a second. I have. Yeah. Let's say here in this scenarios, right, so whenever you give the input to the system, in this case, the system will train, right? So whenever the system trains on your data, if at all, if we give the wrong output, again you need to go back and in your model you need to put, select the features. So for that it is going to, and as I said, it's going to adjust the features, input features. That adjust features are the weights here. Okay, weight up, we can say like weight optimizations. Okay, so that way, by using that weight optimization, we are going to, the system is going to provide you the correct output or not. Okay, so we are going to calculate the loss functions and all those things. Right, so that is the second thing. That for that one, we are taking the Adam optimizer here. Right, now in this case, what I'm doing is I have downloaded my data set and I have mounted to my Google Drive. So to mount your Google Colab to Google Drive, we need to run these steps. Now, so let's come to the data pre-processing step. For the data pre-processing step, I said, like, we need to resize all the images into one part, right? So to resize the images, okay, we need to train our data values here. I mean, we need to resize all the images here. For what we are doing is just we are taking the image data generator, where it's going to resize all our images into something like share range, zoom range, and what is the horizontal flip, and how to how to convert, what is the how much rescale we need to do. So that is going to be done here. Okay, then to train our to get the trained data which is present in our drive. Okay, so this is drive, this data is present in my drive, which is having the training, as I said, right? So in that, if you see in this particular thing, it is going to say like the data organized in folders like training, testing, and the validation, right? So for that, what we're doing is that I'm taking the training data, I'm mounting from my Google Drive, I'm taking all those data. So here, as I said, right, so we need to rescale the data. And here, resize all the images. What we need to do, you need to get all the images, training images, and our target sizes, we need to resize the images by 300 by 300 pixels. For each batch, I'm sending the 120 images, which is going to convert it to binary format. The class model is binary format. Right? Now, so once that is our training model is done, then there are the two type of things here. One is the classifications are there. One is the normal conditions, one is the pneumonia condition. These are our classifications. Right? So for the classifications, just you need to get, I'm giving the normal as a 0, the pneumonia as a 1. Because let's say if the while after training your model, if you get the 0, that that symptom is the 0 is nothing but the normal symptoms. If you get the value as 1, then you have the symptoms as pneumonia. So this is what the thing. Now, so to get the to load our data for our test generation data. Now what we are doing is we are resizing all the images, because as we are converting the test data, we need to convert the as we are converting the training data, we need to convert the test data also. So that is the reason we are converting in the training data. Now I'm plotting all the images from the both like what is that from the training as well as the from the testing. So these are the images that are available in the data set. Like, so these are all the different types of images that I'm showing here. So then, so these are the images without any pneumonia. So see here, without any pneumonia from the data set, these are the images, and these are the images with pneumonia. Okay, so this is one type of data set I'm taking. Okay, so next. Now let's create the sequential model, our model. So here, as I said, we need to apply the different types of things here, like as per our as per our model, right? So we have applied the convolution model. So here I'm taking the convolution 2D network, which is the 16 layers with the filters, with the activation layer, the input as 300. So similarly, I'm taking for the remaining all the data, like remaining all those things I'm taking. Now this is my sequential model. Okay, so which is a sequential model, the output step and the number of parameters I'm taking here. So once you train your model, now I'm going to fit our model in, I'm going to fit the data set onto this model. So my training data is available in the training generation, and applying the number of epochs as a 50 epochs. So the 50 epochs here is okay. So the 50 epochs here, the epoch means here, as I said, whenever you got the wrong output, like whenever you get the your expected output and the real output does not match, then you'll get the obviously will get the loss. Okay, so to reduce that particular loss, what the system will do here is it is again go back and it will update the weights. So whenever it is updating the weights at that time, whenever it will update the weights, again it will move forward. So here initially it will provide all the features has been multiplied with the weights, and it will go forward, and finally we'll get the output. If the final output and your expected output both are having the different loss rate, again you have to do the reverse. So go back. So forward and the backward propagation. One forward and one backward propagation will be treated as a epoch here. Okay. So in this case, so now I'm taking the 50 epochs here. 50 epochs, and our value in the training data. I'm fitting my training data here. So it is going with a 50 epochs. Okay, so 50 epochs. If you see here, initially if you see, loss function will be high because only one epoch means always you'll get the So only one epoch means the system has been trained on the one time. So here we got error rate is 0.57. Then again, and my accuracy rate I got it is the 0.75. Right. So in this case, if you see the accuracy, loss function is having the 0.5, which is the 50% of the data. 50% we got, 50%. 50% I got the loss here. Like means what is my expected data and the actual data is both are different here. Like only 50% are matching here. So to to get the actual output, like to get my desired output. So what is my actual output? To get that, again, we have to do the one more epoch. I mean, go and update the weights. So for that, if you see in the model, here we have applied the Adam optimizer. See, for the model compiler, we are applying the Adam optimizer, which is going to, updates our weights. Okay. Then on that, to calculate the loss function, we're taking the binary entropy. Okay, so our taking the matrix as a accuracy matrix. I mean, normally we are going with the accuracy matrix one, right? So in this case, right? So the number of epochs has been 50 epochs. So initially the 50 epoch, and we got the loss function as 0.4. We'll continue the process, right? So we'll continue the process. If you see, the loss function will be reduced to 0.05 after the number of epochs is 50. The accuracy level we got here is 98.16. So in this case, if you observe, in this case, so the model has been trained very well. Okay, so nothing, we have not done anything here. Simply we have uploaded our data. We have uploaded our data and we have uploaded, we have taken the convolution layers and and we have applied the number of epochs on the training model. Right. So this model, whatever the model you have trained here, whatever the model you have designed here, you need to save that model actually. Because in the later point of time, you can use this as your model. Like you can use this as your AI system. So just simply what the people will do here is just they'll design one A by AI model and they'll give the input to that system. The AI model will detect that is that particular is that particular person is having the pneumonia or not, right? So this is what will happen in the in the deep learning process. So up to this portion, we have applied the convolution layer, and we have taken. We have applied the. We have applied the. We have gone to the number of epochs, and we got the accuracy level as 98.16%. Now let's put everything into the what you call pictorial format. So for the pictorial format, as I said, we are going with the matplot libraries. The matplot libraries is going to is going to put all those things in the form of graphs. So what I'm doing here is I'm creating a figure with 15 by 10 here. This is my, let me decrease the size. Okay, so this is my training data and the validation loss. How much loss I got and how much training loss I got. Okay, so in this case, right? So loss, on the X label, like maybe you will understand simply like this is my training loss and the validation loss, which is going to be happened on the legend side here. And title, this is the title of this picture we are taking as a plot.title. And the X label, I'm taking the number of epochs on the number of epochs, and the Y label, I'm taking the loss function. So our training loss and all those things we are printing here, and okay, so we're plotting the number of epochs, how the it is moving when the whenever we give the 50 epochs, how the model has been increased accuracies and the validation accuracies and all those things have been done here. So to save our model, okay, to save our model, simply what we have to do is just simply you need to select model.save. Okay, the model.save will save the data. I mean, whatever the trained model we have designed, that is going to be saved here. Okay. So once your model has been saved, just load your model into your system, and later point of time, okay, so you can use that model and you can mount your you can validate your data. Let's say here I'm loading my I'm saving my model. Sorry, here I have saved my model. That model loading into the new thing here. I mean, again, I'm looking for the other images. So in this case, the same process I need to go through, like I need to go through like image conversions and all those process I need to do. Now I need to evaluate. We need to evaluate on the results, like on this data set, whatever the model we have done. Right? We have to take that model dot evaluate gender, which is going to work on the data. Okay, which is gives the accuracy levels as well as the loss functions. So on this test data, when we applied, we got 92.174%. Let's use one image to check, like, is that image is having the pneumonia or not. So for that, like, we need to I'm loading the image here. I'm loading the image, and what I have to do, we need to resize the image, right? So because this is the essential step for the pre-processing step. Simply whatever we have done as part of our pre-processing in the images, so same thing we need to do here. So we need to resize and reshape the images. Then go with the model.predict model, which is going to predict the image is having how much is that image is pneumonia image or not. Here the prediction shows a 0.01, which is less than 0.5. So our model predicts normal image. So like the problem here is you need to keep a threshold value here. Okay, you need to predict on the model, like you need to keep a threshold value. For our understanding purpose, we're taking the 0.5. Okay, so 0.5 as a pneumonia image. Okay, so if it is greater than 0.5, I'm considering that as a pneumonia image. Else, I'm going to consider it as a normal image. So this is how the things will work. So same thing. See here, we got second image given. So it is given with 0.99%, where it is greater than 0.5, which is considered to be the pneumonia image. So similarly, we need to do for the remaining things. So this is how the deep learning, I mean CNN, works for this medical image classifications. Okay, so the thing here is, you may go with the next level like LSTM. You may go with the RNN and all those things. But basically, what we need to do here is how you are training your model and how you are pre-processing your model is the essential things. Okay, so and most of the people want people what they will do is, whenever they are testing the model, they won't do the pre-processing. You need to do the pre-processing, then only you need to send the image to the predictions. Otherwise, the size of your image and the trained model images are different. So system won't, sometimes if you give this type of if you do this type of mistakes, then it may be causes the wrong output, right? So this is what will happen. This is what we need to take care whenever you're training the system. Sorry, whenever you're predicting the system. So this is about the things. And if you want to try, you can also trial on the same thing on the LSTM. And if you go with the research kind of things here, if at all, if you want to work on this research perspective, okay. So medical images, medical images are good for the research, because you have a vast amount of data, right? You have a vast amount of data where you can freely available data you can download directly, you can work on it. And there are multiple things you can update in the deep learning. Like, let's say, I'll give you a simple example here. As I said, Adam optimizer. What Adam optimizer will do? So the Adam optimizer, you here, there is a notepad. Just give me a second. So let's say this is the CNN architecture. Let's say consider this is the CNN architecture. And some neurons has been converted. Okay. And these layers are connected to here. Okay. These layers are connected. Okay. Like this. I'm saying about the fully connected layer. So whenever you give the input, it's going to take the features, right? It's going to take the features. And the features are going to be multiplied with the weight factors, right? Weight factors, W0, W1, like that. These factors are multiplied with the features and it's going to provide the output here, right? We'll get the output here. If this output is not as per your expectation, like let's say your expected output is 1 and we got the output as 0. Then what we have to do again, we'll go back, as I said, right? We'll go back and we'll do the backward propagation. While doing the backward propagation, system will update these weight factors. So the thing here is you can also work on this weight, how the system weight optimizations are work. So how the Adam optimizer work, how the weights are updated in the system. So that is the one thing we can do. Even you can, whenever you are working with this, if you are able to optimize this weights, same optimizer algorithm you can apply on the LSTM also, right? So, and even you can apply for the RN, where if you go to any type of CNN or deep learning approaches, everywhere the weights are there. Weights plays the major role. So that is what the research works here. Okay. So that is what that is about this medical image classification, guys. Okay, so let me go through these questions. Yeah, I'll share the code, this Colab code, no problem. So, okay, so that's it from my end. Any questions, team? Okay, so just a second. So, yeah, what are the current research gaps for the medical image classification? As it is appear, people have been applied a lot of deep learning techniques on the data set with good performance. Still, if you ask for the research gaps in the medical image classification, still there are no proper models are there. Let's say if we develop a model, we are not guaranteed that that model will give you the exact 100% result. So still there is a gap. So the classification here, and let's say one more thing here is, if one person may have the as I said, like in this particular scenario, we are working on the pneumonia data set. Let's say the same person is having the other disease. If the same person is having the other disease, this particular work, whatever we have done, whatever the model we have done, it's not going to identify that as a pneumonia, other disease, right? So if we club all this classification into one umbrella, one tree, then that will be the good thing. I hope I have answered your questions. Okay, so just a second. It can be applied for any type of images. Yes, it can be applied to any type of images. But you need to take care of the pre-processing step. So whenever you are training your model, right, sir? Whenever you are training your model, you need to keep all that images into one format. That the pre-processing step will take care. Should we train the model both normal as well as the disease? Yes, we need to train the model for both normal as well as the disease images, because if we train only the disease images, okay, if you give the input as a disease image, system will identify that as a disease image itself, because there is no normal images is there, right? So that is the reason we need to train the both. We need to train both normal as well as the disease images, sir. Yes, sometimes it is taken a lot of time to train. Is there any process we can do it easily? Sometimes in between training it is stopped. So, sir, Rashmita, like, so the thing here is, if you are working in the local systems, definitely deep learning will take a lot of time because it depends on our RAM capacity and all those things. So that is the reason I have executed my my programs in the Google Colabs, because it is going to provide you the 12 GB of RAM for me. In CNN, we are implementing convolution and the pooling operations through the neurons. Yes. Okay. So I hope I have answered all the questions. Yeah. Still any other questions? Still we have 3 minutes of time. If anyone have any questions, please feel free to ask. Let me stop the presentation. Can you tell me any specific about the labeling during the training? You're asking like training in the sense, right? Can you tell me any specific about labeling during the training? So without labeling, sir, simple. Without labeling, I'll share the Google Colab. Yeah, I'll share the Google Colab link. No worries. So see here, as you know, as sir asked, like specific labeling. Say like if you don't have a label, it is difficult to identify what type of data it is. Right. So if we don't have a label, that will be considered as a clustering kind of things. Okay. So it is it is impossible without the labeling data. Okay. So we have to have a labels. So there are many data sets in the Kaggle. How to determine which data set is proper to use? Simple, sir. Whichever the data set is having the n number of workarounds, you need to take that data set. Okay. So like you just go to that Kaggle, there is a multiple people are working on it. Whichever the data set is having the multiple workarounds, you just select that. Okay. Yeah. I hope I have answered your question. I'll share the Google Colab. Yeah. Yeah. So if no questions, we are good to wrap up, right?
Yeah. Participants, if you have any questions, please post in the chat box. Sir, there is a question for you.
Can you suggest any site for hyperspectral image dataset? Okay. It is there in the Kaggle, right, sir? And there is a GitHub pages is also available. In the Kaggle, it is last updated one is the June 2022. It is like 4 years back. I think after that there is no updations on the that one. Yeah, there is one data set is available like ScienceDB. Okay, you just go to that ScienceDB data set, sir. There you will find this hyperspectral data set. Okay, that is where you can find. Okay, let me share you that code, that link. Yeah, this one. That's in this data set, you will find the data set related to your ask. Okay.
Yes, sir. There are no more questions.
The guys are here on the team.
Thank you, sir. Thank you, Suresh, sir, for highly engaging and practical hands-on session on implementing deep learning for medical image classification. So first, your step-by-step approach, clear explanation of deep learning concepts and live demonstrations have made complex ideas easily understandable. The way you connected theoretical foundations with the real-time medical image data sets has provided our participants with a valuable practical exposure and confidence to implement these techniques in their own teaching research. We truly appreciate your time, efforts, and willingness to share your expertise with us. Once again, thank you very much, sir, for your
Thank you.
valuable contribution to the FTP. We look forward to your continued association in the future, sir.
Sure.
Thank you, sir. Thank you.
Thank you, guys. Thanks for your time.
Dear participants, please fill the attendance sheet before leaving the meeting. Link will be active till 9:30 p.m.
Thank you all. I'm dropping off.
Thank you, sir. Thank you.
Thank you.